That's nothing surprising. So today we're going to cover DNA structure and mutation. So a bit of a review, right? You guys know A, T, G, and C. Let's dive into that again. Let's talk about how it works. Let's talk about the structure of DNA, how DNA is repaired, how mutations occur, how mutations are identified and repaired. I think it's just Abigail that took my cell biology class. This will be a significantly less deeper dive than we took last semester into DNA structure and mutation, simply because we don't really look at molecular biology all that much in class. So as we all know, DNA, as well as its partner RNA, allows the flow of DNA directly from the molecular structure. When you see A, T, G, C, and any order, you can interpret that and read those instructions, right? It's like reading a book. Right? That's how your cells interpret those things. Now, DNA is the mechanism by which we study genetics, right? We can't look at alleles without DNA, and we can't look at heredity also without DNA. But ultimately, we have to read DNA in some way, shape, or form. And DNA itself, coolly enough, is mostly chemically inert. It doesn't like to react with stuff. The enzymes that degrade DNA are pretty unstable, so they're called DNases. They're inherently very unstable, and they do that because you want your DNA to hang around, right? That's in contrast to RNases, which regulate RNA, obviously, and they're really, really stable, super stable, because you don't want RNA hanging around for too long. So let's talk about a little bit of history. So the history of DNA dates back farther than you think. We like to kind of think of Watson and Crick as being the first, gentlemen to have credit for studying the DNA, but Miescher in 1869 extracted what he called nuclein from the nuclei, as aptly named, from human white blood cells. So very cool. Now he was a chemist primarily, of course. He obviously was dabbling a little bit of biology here, but he discovered it was weakly acidic and it was phosphorus rich. And in hindsight, we go, oh yeah, that makes sense. We know DNA is chock full of nitrogen, phosphorus. And then he did for further chemical analysis, revealed that its major component was deoxyribonucleic acid, or DNA. Makes sense, as we know. But it contains deoxyribose, found in the nucleus, and it is acidic. So that is 1869. So about 100 years before Watson. Almost. In that ballpark. Back in 1923, we had staining of cells that revealed that DNA was localized almost exclusively within chromosomes, with a little bit of it found in your mitochondria or chloroplasts, if you're a plant. Now, when we look at the chemical composition of DNA, it contains 4 distinct kinds of nucleotides: A, T, G, and C, adenine, guanine, cytosine, and thymine. You should know it and love it. Have it tattooed on your own. Not really, obviously. That level of ingraining in your brain there should be for A, T, G, and C. Now, the nucleotides themselves are made up of a deoxyribose sugar. Remember, that's in contrast to RNA, which is a ribose sugar. What does the deoxy mean? Well, it's simply just missing an oxygen from this pentose carbon here. That's all it means. Now, what links DNA together are phosphodiester bonds. You link the 3' carbon here to the 5' carbon here via the phosphate backbone. Nothing ground shaking here, but that's a special type of bond. And DNA nucleotides are what we call monomers. So they're just individual units. We string them together to form polymers. And that level of structure should seem familiar. When you think about proteins, the monomers of proteins are nucleic amino acids. And when you string them together, they form polypeptides, i.e. strings of amino acids. So that level of structure should seem pretty familiar, but being polymers or linked chains of monomers should seem familiar just generally, right? Cellulose, starch, sugars, all those compounds, even synthetic plastics, right? Yeah, synthetic plastics are polymers. So should seem, again, not new, but just as a refresher. Now, DNA is made up of only 4 subunits. Some people argued back in the day that was simply too simple, which I think is interesting in hindsight because I kind of agree with it. Right? If you think about how complex the human, say, nervous system is, or our circulatory system is, how the heck does 4 DNA bases and 22,000 genes control something as complex as the body? When you think about it that way, that's seems kind of weird, right? At least to me, it always seems weird. People thought it was too simple, and some people, crazy enough, still believe it's too simple. Don't know how, but some people do. There is conspiracy theories that there are other bases out there, just as an FYI. protein is made up of 20 different subunits, which clearly says there's more potential for different combinations. But ultimately, even though proteins have more combinations, more subunits, chromosomes just are, interestingly enough, more protein than DNA, but protein is not the way we store information, right? Proteins, as we know, are structural. They're enzymatic, right? Catalyzing reactions. So we know DNA is the bee's knees for transmission and inheritance of information through time. But let's talk about how we got to where we are, because there's a really fantastic set of experiments from the 1920s all the way up to the 1960s looking at DNA and how it actually factors into the inheritance of genetic material. And I look back at this time period and I kind of like fondly look at it because it's like almost like a Wild West. They had no idea what the heck was going on back then, right? These guys didn't know what DNA was and all the stuff we have today, right? But at the same time, we just landed another probe on Mars, which is insane. And we just developed a vaccine in 8 months, which is also insane. So I look back on this and say, wow, must have been so cool, but at the same time, there's way cooler stuff happening. Right. But let's talk about Frederick Griffith, because he's a microbiologist, and he's one of the early pioneers of trying to figure out which was the inherited material, was it protein or DNA? Remember, protein is a huge component of DNA, so they didn't quite know, protein or DNA. So Griffith in 1928 did a couple experiments. You probably heard of this one. It's a pretty classic experiment, but Griffith worked on Streptococcus pneumoniae. So as its name implies, it's one of the causative agents of pneumonia, as you can see from its species name. Now, pneumonia is just kind of a broad class of organisms that can cause a pretty serious lung infection. This just happens to be strep pneumoniae, just as a note. You also recognize Streptococcus as in strep throat. So if you ever had strep throat, you can think strep bacteria. Now, strep pneumoniae has two distinct phenotypes. Smooth strain, which we note always as the S strain, which is the virulent strain, and the rough strain, or R strain, which we denote as the non-virulent or the asymptomatic strain. Now, the R cells, as you'll learn, could be transformed by the genetic material from heat-killed S cells. What is transformation mean in bacterial context? Well, it just simply means when a bacteria uptakes genetic information from the environment. Water. So let's talk about what Griffith did, but this is what we're looking at. The rough colony of strep, smooth colony, smooth being pathogenic, rough being avirulent, right? Not disease causing. As a note, smoothness is a pretty common trait of pathogenic bacteria. Being smooth helps you evade immune systems.
What was it that you said just now with the rough and the smooth? What's the difference?
One is pathogenic, so the rough is, the smooth is pathogenic, and the rough is non-pathogenic. And we can look at a schematic of Griffith's experiment. So we took the virulent S strain, injected it in some unfortunate mice, and they died. As you'd imagine, if you injected pneumonia directly into your blood vessels, you would probably die from it as well. And that's it. Then he took the rough strain, injected it into mice, and they lived. As you'd imagine, if you inject a mouse with non-pathogenic bacteria, it's probably not going to die, and that's what happens. So that's cool. And then he took the virulent S strain, he heat killed it, and he had just their cellular components, and he injected that into mice, and they in fact did not die. So nothing surprising, right? Pathogenic bacteria kills you, non-pathogenic bacteria does not kill you, and heat-killed bacteria that's being killed simply cannot kill you. However, he did a little funky thing. So he took his pathogenic bacteria, he heat killed them, and he took his non-pathogenic bacteria, still living, combined them together, injected them inside of a mouse. The mouse died. And then he harvested tissue from the mouse, he re-isolated his bacteria, and what did he find? The living S microorganisms. So the pathogenic ones. So what's going on here? Well, this is what transformation is. The picking up of DNA from the environment. Oops. Where we have this organism, well, it was originally the non-pathogenic version. It picked up DNA from the pathogenic version, which is beneficial to it in its current environment, i.e. a host, causing it to essentially mutate, right? Change forms, thus becoming the pathogenic one and being deadly to this mouse. But at the end of the day, Griffith didn't know quite what he was talking about, right? This is 1928, right? We didn't have Chargaff or Franklin or any of those pillars of DNA yet. However, he did know something about this cellular mixture was causing the switch from non-pathogenic to, in fact, being pathogenic.
So the little purple ones are the ones representing the smooth or the rough?
Rough.
The rough.
R and S being smooth, R being rough.
Okay. So the rough combined with the heat killed smooth?
Yep.
It did kill them all? How was that?
I just told you. This organism picked up material from the lysate of this cell, thus transforming it into a pathogenic version. Now, at the time, Griffith didn't know, right? Griffith said, okay, I have the cellular components from this dead bacteria. I don't know what's causing this bacteria to switch from this to this, but something in there is causing the switch. He termed this transformation because it was transforming R back into S. And the idea is it's picking up something. Now, we know this now to be DNA, but in 1928, they had no idea what it was. So, fast forward, almost 15 years, Avery, McLeod, and McCarthy, they actually physically identified the transforming principle. So basically, they provided evidence, and I like this transforming principle. But they basically purified the cellular extract. So Griffith was basically just taking all the bulk parts. They were purifying the actual physical extracts and injecting it, and thus developed a more concrete principle of the transforming principle. Nothing crazy. They didn't really learn anything new, but they just defined the transforming principle. And what they actually did was take the purified transforming principle, i.e. the pathogenic organism's materials, and they actually treated it with a couple of different things. So they treated it with essentially either enzymes or centrifugation or chemical analysis. And interestingly, they treat it with a protease, which obviously, as its name suggests, kills protein or destroys protein. So they treat it with a protease, destroying essentially all the protein, not all the protein, but essentially all the protein. It's impossible to fully get rid of any of these components with just an enzyme in a tube, but it was pretty close. So they took this whole cell lysate, which they called the transforming experiment. The transforming principle. And again, all that is just the heat killed components from an S or pathogenic Strep pneumoniae. Protease killed the proteins, which they reintroduced to R cells, which led to the transformation of those S cells. So clearly protein wasn't the causative agent here, right? This wasn't what was transforming rough, i.e. non-pathogenic, into pathogenic. Then they treated with RNase, which destroyed the RNA as its name implied. They introduced it to the R cells and the S cell. They still were transformed into the S cells. So clearly RNA is not the causative agent here. Then finally, when they treated with DNase, i.e. destroying the DNA, again, it's not perfect, but it was clearly enough to see a result. They introduced it to the non-pathogenic R cells, and there was actually no transformation that occurred, which implies to these folks, and obviously to us now, that DNA has to be the transforming agent here. They also tried some other stuff. They got rid of fats. They also did some physical and chemical analysis, and all their results pointed to DNA being the only agent here. And again, in hindsight, we go, duh. But back then, lawless 1940s of science, they did. So DNA clearly has to be what's going on here, but not everyone was actually convinced by this data. They just weren't. People were still skeptical, and as you guys probably know, a good scientist is skeptical. So you would imagine like this is pretty concrete evidence here, especially if you have a big data set to prove it. So that's cool. But bacteria are a bit, they're a bit complex. So we take it to a simpler system, which are our viruses. So viruses that tag only bacteria, or infect only bacteria, I should say, or are bacterial phages, or just phages for short. And they have this wacky and wild moonlander shape, which is one of the reasons I think they're pretty cool. They look like a moonlander to me. Now, phages, as you guys may have picked up from learning about COVID, or if you bothered to learn about COVID, that is, they're just basically proteins, maybe a little bit of the cellular membrane. It's an envelope virus like COVID is. And they have their nucleic acid, DNA, or RNA. COVID is an RNA virus. The vast majority of phages being DNA-based virus. Now, the interesting thing is, phages, not all viruses, but phages, contain equal proportions of protein and DNA. This provides a great idea for how to disentangle, is it protein or DNA? Now, clearly, this previous slide should tell you it's DNA, but again, people still thought protein was the more complex form, thus it was most likely to be the agent of heredity. And we can use these phages to detect what's going on. What material do we need? Protein or DNA to encode for a brand new phage. Because remember, all a phage is a parasite. All any virus is a parasite. They have their DNA or their RNA, single-stranded or double-stranded. They inject it into the host, which then reprograms the host to make more viruses. That's all a virus is, just a parasite. So phages are great. Now, when we look at these T2 bacteriophages, so it's just their name, T2, they infect E. coli. Now, these bacteriophages have very few genes, but they're obviously able to replicate themselves, right? That's a hallmark of life. You have to be able to reproduce. And that's all life's purpose, right? Now, the way any phage works is the virus hops onto the cell. It recognizes typically a receptor, sugar receptors are by far the most common targets. So, for instance, HIV targets the sugar receptors on your white blood cells. So, any phage or any virus attaches to the host, injects their genetic material. The phage then begins to replicate its DNA or RNA, and thus using that to make brand new viral proteins.
Does the phage die off after it injects it?
It's not really living to begin with.
So does it just fall off?
It's not like malaria, right? It's not like a living organism. It's just a piece of protein and DNA or RNA. So this just kind of falls into the ether.
So it's kind of like, I don't know if this analogy makes sense, but it's kind of like landing on something like the plane or the moon or something, and then letting us come out and then just like the planes. I don't know.
I mean, they left lots of parts of the original lunar lander there, so yeah, it's kind of like that. Yeah, so the original phage just goes bye-bye. But the idea is once you eject your genetic material, well, you have all the instructions to make thousands to tens of thousands of more viruses. And that's all the virus really wants to do. That's all.
I was just trying to make an analogy.
Yeah. And that's all life. You really don't really care about what happens to this original one. It's one viral particle. We don't really care about him. We care about what's coming out the back end. Yeah. But again, the phage injects its genetic material, reprograms the host to make new parts of the phage. They're just basically making more protein. Right? The phage is just nucleic acid and protein. That's all it is. Then they assemble the phage particles, which again is encoded partially by the phage's genome. They also rely on the host genome sometimes, which is kind of a cool thing. And then they lyse the cell, causing it to burst, releasing new phages to then go on in to affect new host cells. Again, this could be host as in your body, lung cells, liver cells, whatever. Or in the case of bacteriophage, it's going on to affect more bacteria. So Percy and Chase tackled phages to concretely, essentially settle the argument here. Is it protein? Is it DNA that is the inheriting agent? And they did a pretty cool experiment. Now, what they did is they had T2 phage. They had two different groups of them. One that was infecting E. coli growing on isotopically heavy phosphorus. So it's P32. So it's, phosphorus is normally P31, if I'm not mistaken. This is P32. And then they had E. coli growing on S35, so slightly isotopically heavy sulfur. Sulfur is normally S34. That's what I know off the top of my head. Now, why did they do that? Well, we know DNA, based upon that experiments done in the 1830s, that it was huge amounts of phosphorus. And the reality is there's basically no phosphorus in proteins, and they already knew that. So they could look at this and say, okay, well, protein tags DNA, not protein. Conversely, S35, well, why do we want to use S35? Is there sulfur in DNA? Is there sulfur in DNA? Nope. It's a straight question. I don't ask straight questions. No, there's no sulfur in DNA. But is there sulfur in protein? So yes is the answer, but where is it in protein? There's only one amino acid that has it, amino acid called cysteine. So cysteine being the only sulfur-containing amino acid. Well, methionine has it too, but methionine is a start going on, so it's not very abundant. So we know that sulfur is only found in protein, it's not found in DNA. So you can label isotopically heavy DNA and heavy sulfur. I'm sorry, heavy protein by looking at phosphorus and sulfur. Pretty cool experiment to set up. Now, why would we label it heavier? Well, the reality is we can actually let the virus do its thing. And because they're isotopically heavy, they're actually slightly radioactive. Phosphorus, you guys might know, is radioactive. S35, also radioactive. So you guys, phosphorus will be radioactive. Anyways, so that's cool. They're also slightly heavier, right? 35 is heavier than 34, and 32 is heavier than 31. So with those two things in mind, you can detect radioactivity, and you can detect things by weight. Slight differences in weight, but you can detect them by weight. So the idea being is if you have this that targets DNA, that targets protein, when you produce new viruses, they'll either have heavy DNA or heavy protein. It's pretty ingenious. Then you let them go on to affect a bacterial culture that's not grown in heavy sulfur or heavy phosphorus, and you look for where the radioactivity is in the brand new generation. Is the radioactivity inside the cell, or is the radioactivity hanging out on the ghost viruses, right? Because if it's carried in the protein, well, it's going to be on these dead viral corpses. If it's carried on the DNA, well, it's going to be in the DNA that's been injected inside the bacterial host. So it's a pretty ingenious experiment. Now, in hindsight, we know what the answer is. We know that we find the radioactive DNA inside the cell, and the radioactive protein just kind of hangs out, right? So this tells us that because the protein doesn't go into the cell, it is simply not the way the virus infects a bacteria, which means that the way a virus does its thing, the way it carries its genetic information, is actually not protein, but DNA. So a couple really cool experiments to set up how we figured this out. Because again, we take it for granted now, but back in the day, we had no flipping idea. Wild West, baby. So basically, I just talked about this, so we'll go on to the next one. So now that we know that DNA is the way we inherit information, right? We know alleles are carried on DNA. It's the mechanism by which heredity and genetics work. Let's talk about its structure. So as we mentioned, DNA is a polymer. It's comprised of nucleotides, deoxyribose sugar, phosphate group attached to a nitrogenous base. Now, there are two different classes of nitrogenous bases, are purines, which is a 2-ring structure, or a pyrimidine, which is a 1-ring structure. Fun. And this is what we have. We have the deoxyribose sugar. The deoxy part comes from here. There's no oxygen attached to this part, just the hydrogen. We have the phosphate group PO4, and then we have our purines, which are adenine and guanine, and our pyrimidines, which are thionine and cytosine. So nothing new, but just as a reminder. Now, we actually start to build these, and we start to get some terminology. So we have nucleosides, which I always forget is a thing until I have to talk about it. Because I'm always just thinking nucleotides, but nucleoside is a thing, which I actually just don't like the way it sounds, but it is, which is simply the 5-carbon sugar attached to the nitrogen space. So that's a nucleoside. Then we step up to a nucleotide when we tack on the phosphate group, leading us to our and then that's our purine. Then we have a pyrimidine, again, phosphate, 5-carbon sugar attached to a nitrogen base. I know, exciting stuff. Now, DNA, like proteins, have directionality to them, right? When you read a book, you, at least in English, in most other Latinized or Germanic languages, you read from left to right, right? You don't read from right to left, and DNA is the same way, right? Now we link our DNA together in a 5' to 3' direction, with the 5' being the phosphate group, and the 3' group being the 5-carbon sugar, and you always link 3' to phosphate to 5', 3' to phosphate to 5', over and over and over and over again. That's how you join our DNA. Nice and simple, no other variations on the fact whatsoever. There's only one way to join DNA. And remember, when we join it this way, 3' to phosphate to 5' carbon. This is a phosphodiester bond. If you want to remember what an ester is, I just Google what ester looks like. And you'll see why it's a phosphodiester bond. And remember, DNA has directionality, 3' to 5' direction. You go this way, not this way. Now, that's what we knew by the early 1950s. 1952, one of the most badass ladies in all science, Rosalind Franklin and Maurice Wilkins, obviously not a badass woman in science. They solved the X-ray diffraction pattern of DNA. So in your textbook, if you want to take a peek at it, it's 6.8, figure 6.8. I don't know if you guys read your textbook or not, just as a note. You can also just Google Rosalind Franklin X-ray crystallography of DNA, and you'll see the picture. I didn't put it here because the picture you guys have seen a bunch. It's just the X. So they resolved a couple important parts. So DNA's helical structure has a 20 angstrom diameter. So remember, angstroms are teeny, teeny, tiny. It's what, 10 to the minus 12 meters per angstrom, if I'm not mistaken. Might be slightly bigger than that. I never can remember the exact number. The spacing between the repeating units, so our sugar, phosphate backbone, DNA nucleotide, that sort of thing, is 3.4 angstroms. There's a helical turn that's complete. So if you complete 360 every 34 angstroms. Pretty cool. Yep.
Do you want us to remember the scientists and the year and all that? Or...
I think you should just remember them because they're really important, especially Rosalind Franklin.
No, I'm just like on the test. Like
I don't ask. This is not a science issue. I might reference their work and what they interpreted from it, but I don't expect you to remember, oh, this person was this. I expect you to remember the
Okay.
But it's important to put all this in the context of broader history, right? Because the problems that Rosalind Franklin and many others at her time, Marie Curie, and so forth, were dealing with are still persistent in science today. So, and just general society. So it's important to understand them as people and how important contributions they made. She should have won a Nobel Prize, as should have Chargaff as well. But they didn't because, at least for her, because of problems in science in his women, which again still persistent. They're less severe, but they still persist. So it's important to have this background knowledge about how awesome Rosalind Franklin is. Now, they also had some pretty detailed knowledge of the chemical constituents of DNA, so they helped identify the A, G, T, and C. Right? And also, they noticed the deoxyribose and the phosphate. Now, along came Erwin Chargaff. He came up with our complementary DNA base pairing not too long after. I think it was in 1953. He always found that A was always, he did all this chemically, just as a note, but he always found that A was in a 1-to-1 ratio with T, and G was always in a ratio of 1-to-1 with C. A with T, G with C, A with T, G with C. And it's actually kind of cool. So you can look at the A to T or the G to C ratio in all organisms, chemically defined. And you'll notice that the percentage of bases in E. coli and all these different organisms, all the way up to humans. And you'll notice that the ratio comes out to be about 1-to-1 every time. It's not perfect. It's really hard to get that exact ratio back in the day, just chemically, but you can do it now with DNA sequencing. It's not perfect, but it's pretty good. What you'll notice is that the G to C ratio and the A to C ratio, they're again 1-to-1, but you'll also notice that the proportion of A's and T's varies pretty dramatically. Right? E. coli's got 24% A, whereas C. elegans, so just a teeny tiny worm, you can barely see with your naked eye, has 31% A. That's actually kind of an interesting phenomena that you can have variable amounts of DNA bases. There's a cool phenomena that more closely related you are, the more similar your essentially your base pair composition is. So you can look at Mus musculus and us, about almost identical percentages of A and T and identical ratios of G and C. It's kind of a cool thing. It's evolutionarily conserved. It implies there's a common ancestor. So fun fact, you can actually use these ratios to actually see, okay, we have a stretch of the genome that doesn't quite fit these ratios. It probably came from something else. So it was essentially horizontally transcended. So kind of a fun thing. All right, so complementary base pairing. So this is Chargaff's area. They consist of hydrogen bonds. So remember, hydrogen bonds form between either Well, they form between changes in partial electrical charges. So as you may recall, when a nitrogen bonds to a hydrogen, well, that creates a slight dipole, i.e. the nitrogen becomes slightly negatively charged and the hydrogen becomes slightly positively charged. Why is that? Well, chemistry will tell you there's a slight difference in electronegativity here. Nitrogen just wants those electrons more than hydrogen. So it creates this slight dipole, this slight partial charge. Oxygen does the same thing with carbon. Oxygen is more electronegative than carbon, so oxygen becomes partially negatively charged. So anytime you have a partial negative charge interacting with a partial negative, interacting with a partial positive charge, you get a hydrogen bond. Now, hydrogen bonds can only form between oxygen and hydrogen and nitrogen and hydrogen. That's pretty much all it has. So but ultimately, this weak electrostatic charge is what holds DNA together, primarily. Now, we know these hydrogen bonds exist, but there does seem to be some effects of water here. So DNA is slightly hydrophilic on the backbones, slightly hydrophilic here. So it seems to be that hydrophilicity also contributes to the holding together. I know it doesn't say that here, but that's a brand new hot off the press research item that came out last year. So this is what we have, hydrogen bonds. And you'll notice that our purines bond 3 hydrogen bonds, and our pyrimidines have 2 hydrogen bonds. Thus, you can infer purines are stronger than pyrimidines. And you would be right. Interestingly enough, when you look at thermophilic organisms, whether it's fungi, algae, or bacteria, or archaea, they have more G and C than they do A and T. Why? Because G and C are stronger than A and T are. Kind of cool. So this leads us to Watson and Crick. And they're the guys that essentially took all this information, so this 80 years or so of material that was gathered, and they have that canonical picture of them too, looking like big nerds pointing at a model of DNA, which we know and love today. So they're the ones that put things together. So they found that DNA strands were anti-parallel. And what does that mean? Well, as you may or may not be able to see on this figure, it goes 5' to 3' on this strand and 3' to 5' on this strand. So they just simply go in opposite directions, right? DNA has that polarity to it, right? It has that direction. So they're just simply in opposite directions from one another. On the outside, we have the sugar phosphate backbone. As you can see, it's P and S here. Again, stuff we guys have heard before, but just reiterating it. Whereas the base pairs come together in the center here, where they do their hydrogen bonding / interaction with water. And that's our DNA. So we have the 20 angstroms set apart by our friend Franklin here and Wilkins, the 34 angstroms for a complete turn. And we also have a couple other things here. We have the minor groove here, and then we have the major grooves here. So big openings in the DNA, little openings in the DNA. That's all. So just some anatomy of the DNA. In terms of what's important, you guys want to keep down the ideas of anti-parallelity. So, 5' to 3' and then 3' to 5' in the opposite direction. A with T, G with C, and all the standard bonding, all the standard information about DNA. I'm less concerned about the size of DNA and the major and minor grooves, but that is a thing you should put to memory at some point in your life. Now, DNA also has 2 distinct forms. Now, you guys are commonly used to seeing the B form of DNA, where we have this right-handed helix, so it essentially turns in one distinct direction. And then we have the Z form of DNA, which I hate looking at, especially this figure right here. I hate looking at this figure so much. But this is the Z form of DNA, which turns the exact opposite way. This is relatively rare, not particularly common. We're not going to discuss it more for this class at all, but just it bears mentioning the thing. But the B form of DNA is by commonly the most common way we see DNA. It's like, I think it's like 95 or 97% of the time it's this, and 3% of the time it's this. I just make mention of it. You want to look up the nitty-gritties of it by all. Now, DNA, it's double-stranded, but DNA itself can be single-stranded as well. DNA can be linear or it can be circular. So when we were talking about the bacteriophages, they had a single linear piece of DNA. When we were talking about the strep pneumoniae, it had a single circular piece of DNA. When we look at other types of Genetic material, it's combinations. Right? Chromosomes are linear, right? But your mitochondrial DNA is circular. So it's kind of depends. And we can look at just some micrographs of DNA. I always wanted to look at electron micrographs. Now, as I mentioned, DNA stores our genetic information. So when we're talking about traits or we're talking about anything genetically related, well, DNA is the mechanism by which this occurs. Right? Unless we're talking about like a virus, where RNA could be the mechanism. But we don't really think about viruses in the class. We're mostly thinking about eukaryotes and bacteria. Now, we have to be able to read this DNA. Because remember, DNA is the way we pass on material to the next generation. It's our storage bank, right? It's our book. Now, if you wanted to use a book, you're not going to just rip the pages out of your book, right? We could, but that would just be a waste of the book. Well, DNA is the same way. You don't want to just rip genes out of your book, your DNA, right? You got to make copies of it, right? Which brings us to my also awesome analogy of what transcription, right? Where we have DNA can be synthesized by an enzyme into its counterpart, RNA or ribose nucleic acid. Moving down that moving down the central dogma of biology here. Now, the vast majority of DNA is unreadable when it's bound together in double-stranded form. It needs to be single-stranded to be read. That being said, there are some DNA binding proteins and tags you can stick on DNA to regulate gene expression, which you don't have to unfold the DNA at all to look at. But if you're going to access the information in DNA, you got to unwind it. Right? If you're going to read a book, you got to open the book. Okay. And RNA is a really fantastic molecule. It's really cool. If you haven't dove into the biology of RNA, like really nitty-gritty of RNA biology, it is one of the most fascinating molecules out there. It's inherently very unstable, but it also is catalytic. And it's not just single-stranded, as you may like to think. It has all sorts of capacity to take hairpins and turns and form secondary structures with itself. So it's a pretty cool molecule out there as a whole. It doesn't form double-stranded things, not in the sense of DNA, but it can form, as you can see here, some binding with itself. Not for a whole stretch of it, but it can bind to itself for a little bit. So let's talk about the Watson and Crick model of DNA replication. So how do we keep our DNA going from one generation to the next, right? So we talked about meiosis previously, right? We talked about mitosis. Part of that was just actually physically replicating your DNA. You can replicate them chromosomes if you are going to make gametes. Right? You need a full suite of chromosomes. So now Watson and Crick proposed that they unwound the double helix, exposing bases on both strands. Each strand then acts as a template for the synthesis of a new strand, and the new strand forms by insertion of a complementary base pair. And that should seem pretty much correct to you, and you'll be right. That's basically how DNA works. And the result was a single double helix becoming two identical daughter double helixes. And again, that should sound perfectly correct, because it is, right? What they proposed was correct at the time. Now, how it was done, they didn't quite know. And I say how it was done, which leads us to three methods that you guys have probably seen at some point in your biology careers. DNA can be replicated in one of three distinct ways. Semi-conservatively, which is the Watson and Crick model. Conservatively, which is the parental strains the parental double helix remains intact, whereas both brand new strains are essentially unique. So you have one DNA strand that's old and one DNA strand that's completely new. And then you have the dispersive, which is the wacky and wild one, where both strands of the daughter helix are essentially contain both a mixture of original and old DNA. So again, it's kind of wacky and wild model. So this is what we know to be true, semi-conservative, half and half, right? Whereas conservative, which would be whole and whole, right? Just completely brand new, old and old. And then we have the wacky and wild dispersive, which is a patchwork, like a mosaic. Think of it like a quilt of DNA. So which is it? Right? We know the answer to this now, but how did they figure it out? How did they prove what Watson and Crick put forth? Well, the reality is it took a couple of years. So Messelson and Stahl. We'll see Messelson in a few slides again. Pretty cool scientist. Now, what they did in the late 50s, they separated pre-existing, what we would call parental DNA from newly synthesized daughter DNA by molecular weight. So one was slightly heavier than the other. So again, you could do isotopic. Then they grew E. coli containing 15N nitrogen, so heavy nitrogen, then switched it to media containing 14N or regular nitrogen. So heavy nitrogen and normal nitrogen. Again, doesn't seem like a lot, right? Nitrogen is 15 grams per mole. So in its isotopic state, and 14 grams per mole in its non-isotopic. So not a lot. I know it's 1 gram, but with scientific techniques, that's easy to separate. 1 gram is a lot. So now what they did is they purified DNA from cells and they subjected it to what we would call equilibrium grade density gradient ultra centrifugation. And to break that down, ultra centrifugation means they spun it really, really fast. So when you guys are in the lab with me, and you use the centrifuge back there, and you spin it at 15,000 times per second, that's pretty slow. They're talking about spinning it at hundreds of thousands of times per second. That's what ultra centrifugation means. Technically 50,000 plus times per second. Density gradient means there is a gradient of gel, as with gel electrophoresis, as you guys will do in lab this week. You'll notice that things separate based on size. And you can also use density gradient to separate things based upon mass. With things that are lighter moving farther, things that are heavier not moving quite as far. Which again should sound exactly like gel electrophoresis or SDS-PAGE or any other gel-based technology. And equilibrium just allows them to come to equilibrium, right? Come into a steady state. So that's that massive stuff they did, but they did it with a chemical called cesium chloride, which forms that density gradient, putting the highest density at the bottom of the tube and the lowest density at the top of the tube, which again helps separate things based on mass. And DNA itself will form a tight band at a position where its density is equal to the cesium chloride gradient, right? So you think about when oil in water, or that's a bad example. Think about when you put like, if you took, say, a rock and you threw it into a test tube. Well, the rock is more dense than the water, it sinks to the bottom of the test tube. Conversely, if you put something, say, like gelatin is much less dense than water. Well, the gelatin floats to the top, right? It stays on the top of the water. So the same idea. Heavier things sink, lighter things stay afloat. Which should tell you, based upon this part here, heavy and normal, well, you should be able to detect the difference between heavy nitrogen and normal nitrogen with this technique, and you'd be absolutely right. Heavy nitrogen should be lower, lighter nitrogen should be higher. And that's exactly what they saw. So this is what they did. So they had control E. coli, because all good experiments need a control. And all the experiments we've talked about thus far had a control. We just haven't made mention of it. But this, in case, it's actually really important to see the control here. So their control experiment, they grew E. coli only in 14N nitrogen for many, many generations. They extracted the DNA from the cell, they centrifuged it, and they found a single band near the top of the tube that was just 14N and 14N. That's cool. Then they grew E. coli for many generations in 15N media. They extracted the DNA from the cells, they centrifuged it for a while, and then they found DNA bands of 15N only at the bottom. And it says 14N, 14N, because remember, there's double strands. So 14N is one strand, 14N is the other strand. Second experiment, they took cells that were allowed to replicate once to produce a generation of daughter cells. They extracted the DNA, and they centrifuged it, and they were growing them in both 15N for a little bit, and then they switched to completely growing in 14N. So the DNA, in theory, should be 50-50. So they extract the DNA, they centrifuge it, and they find a band of 15N and 14N smack dab in the middle here. And then the final part. So they took these cells after 30 minutes. They allow them to replicate not just once, they saw it here, but replicate a second time. They extracted the DNA, they centrifuged it, and they found two distinct bands. One that was 14N, 14N, one that was 15N, 14N, which implies the model of semi-conservative replication. Again, really ingenious way of doing this, getting the bacteria to uptake heavy nitrogen so you can track how its DNA is formed. They knew how E. coli grew. It took E. coli only 30 minutes to replicate. So they know after 30 minutes, only one round of replication would have occurred. So they can use that to say, okay, well, it's a mixture of the two. And then they can say, okay, given that it's in a brand new generation here with only 14N nitrogen, well, it has to produce only 14N nitrogen for that second generation. But you can see still the remnants of the first generation right here. So it's a really cool experiment. Yeah, just using isotopes paired with biology to understand how DNA is semi-conservatively replicated. Again, where you have two strands that are made, half is old, half is new DNA, confirming what Watson and Crick hypothesized. So that's not all. Now, Alex Kornberg and many others, but he gets the credit for it, worked out the biochemical aspects of DNA replication in E. coli. Now, they use energy for DNA synthesis, and you guys should probably make note that you guys should probably know that the energy in cells comes from ATP, adenosine triphosphate. Right? That's the energy currency of cells. Now, DNA polymerase catalyzes new phosphodiester bonds. So DNA, polymerase, poly, as in adding polymers, ACE being enzyme. So it's an enzyme that catalyzes adding new polymers of DNA together. Right? That's all that's telling you. And it has a highly coordinated process. It's separated into two distinct stages. First being initiation, where a protein simply opens up the double helix, because remember, DNA is useless if you don't crack it open, and prepares it for complementary base pairing. And then you have the elongation, where proteins, i.e. DNA polymerase, catalyzes the creation of brand new DNA sequences from an original DNA template. So let's take a peek at this. We have DNA replication now. DNA replication, as you remember, always proceeds in a 5' to 3' direction. That's the only way DNA replication can happen. Now, what does that mean practically? Now, you'll notice that the polymerase here in yellow, oh, that's right, the polymerase isn't actually here. You'll notice that the polymerase, which is denoted by this orange arrow, well, it's going from 5' to 3' on the new strand. But DNA polymerase can't do anything without a template strand. So DNA polymerase adds from 5' to 3', that directionality we discussed, but it's copying from the 3' to 5' end on the old strand. That's the part people always get confused. 5' to 3' on the new strand, 3' to 5' on the old strand. Something that people always get confused at. Doesn't matter where I've ever taught, people always forget that part. They think, oh, it's 5' to 3', so it has to go this way to this way. No, it's 5' to 3' on the new strand. I'm serious. It doesn't matter where I've taught, Northeastern, BU, here, Rock Creek, Greek College. Doesn't matter. Everyone always gets that. And I won't lie to you. Back in the day, I also did. So, makes you feel better. Your own professor gets confused. Actually, I get confused a lot. But that's not uncommon for us. Now, there are 3 strict requirements for DNA polymerase to do its thing. It needs 4 DNTPs, so dinucleotide triphosphate, ATG and C. That's all dinucleotide triphosphates referred to. Now, when we set up PCR last week, or when we set up PCR later in the semester, we added synthetically created DNTPs to our PCR mixture. Because why? PCR is basically just DNA replication. Now, these DNTPs, they're for incorporation into a brand new chain, and they also are for energy, right? The phosphate bonds on these DNTPs, again, dinucleotide triphosphate, right? Triphosphate energy, right? So they have energy and for incorporation into the base. Now, how do we create single-stranded DNA? It has to be unwound first, so you have to have DNA gyrase to relax the DNA double strand, and then you have DNA helicase to simply unzip it. Again, it's like unzipping like a zipper. So let's talk about this. So first start with initiation. So an initiator protein binds to what we call the origin of replication. Now, the origin of replication, as its name implies, is the starting point of DNA replication. Now, in a eukaryotic chromosome, which is relatively large, well, there's multiple origins of replication spread across your genome, spread across your chromosome. But for our microscopic organisms, our bacteria and archaea, there's only one origin of replication. But a protein, this initiator protein, has to physically bind to the origin of replication, and the origin of replication is simply just a conserved set of DNA sequences. That's really all it is. Just a few, it varies depending on the organism, but the origin can be a few bases long to a few hundred bases long. It's just a conserved piece of DNA that says, hey, start here, help us. DNA helicase unwinds the helix. Again, unwinds as in, right, like a zipper sort of aspect. And then the replication bubble forms with a replication fork at both ends. So we have the fork and the fork. And then a single strand binding proteins do keep the DNA open. Now, DNA's natural tendency, because it's held together by spontaneously breaking and spontaneously reforming hydrogen bonds. Once you break DNA apart, it wants to go right back together. So DNA naturally wants to do that. So we have SSBP, or single-stranded binding proteins, as you can see. Well, sort of. I hope you can kind of see them here. Kind of hard on the board. But that's what's keeping the DNA strand from essentially closing in on itself. Then we have primase. So you should recognize it's an enzyme because it ends in ASE. This synthesizes RNA primers. Now, these are anti-parallel to each template strand. So you'll notice that we're on this strand, right? We're going 5' to 3'. So they're anti-parallel. They're 3' to 5'. Same thing for here. And they're synthesized on a very short section. RNA primer length varies. It shows you here it's 2, but they're typically between 7 and 15 bases. It just depends on the organism. They vary. But the whole point of RNA primers, they are RNA. So it's A, U, G, and C. But they're basically designed very much like the origin of replication. They say, hey, come over here. And what do they say come over here to? Well, they say come over here to the DNA polymerase. The RNA primers, remember, are saying, hey, here's where you're going to start here's where you're going to start synthesizing new DNA. And they're there. So that's what recruits our DNA polymerase. Now, when we're doing PCR together in lab, we have DNA-based primers to recruit DNA polymerase inside of a test tube. So it's the same idea as here. So just kind of, that's a nice parallel between lecture and lab here. I like it when that happens. Doesn't always happen. Now, DNA polymerase 3. So in this case, there are multiple classes of DNA polymerase. So E. coli has 4 classes of DNA polymerase, with the 4th one being the coolest one. You can Google DNA polymerase 4 for E. coli if you want to know why it's so cool. But DNA polymerase 3 is by far the most common type of polymerase that synthesizes new DNA strands. And what is it doing? All it's doing is catalyzing the formation of phosphodiester bonds. That's really all it's doing. So it's polymerizing a brand new strand. So again, what we've talked about thus far, we have replication forks here, and there's technically one going to be down here as well. We have the single-stranded binding proteins holding here. We have helicase unzipping. And then we have DNA polymerase that's then bound to the RNA primers. It is synthesizing in the 5' to 3' direction. But as you guys know, DNA runs antiparallel, which means there's one DNA strand that goes in one long zip, right? It's the leading strand because its strand that it's focusing on is in the 3' to 5' direction. Thus, it can go simply, it's 5' to 3' direction all in one go. Which leads us to the lagging strand, which should tell you, well, why is it lagging strand? Well, because it has to synthesize DNA in bits and pieces. Right? So the RNA primer has to come in from primase. DNA polymerase binds to it, and it has to only synthesize in the 5' to 3' direction. But you'll notice that the strand is not in quite the right orientation here. So what ends up happening is the primase has to synthesize a new primer downstream. The DNA polymerase has to jump back and synthesize again in that 5' to 3' direction, creating what we call Okazaki fragments. This is actually a big mystery up until the 80s. It was the 80s. When a Japanese man, obviously his last name is Okazaki, found out this is how DNA replication actually works. Because again, we have this anti-parallelity here. I don't even think that's a word, but our DNA strands are anti-parallel. Right? They inherently create this problem all on their own. And why do they create this problem? Well, it's because DNA polymerase can only read in one way. I can't read a sentence from right to left. I have to read my sentence from left to right. Because if I read it from right to left, it doesn't make sense. I can piece it together, but you know, I'm not an enzyme, or my only function is to go from A to B, not B to A. As I mentioned, the leading strand has continuous synthesis of 5' to 3', so it's heading in the same direction as our, what again we call a replication fork, whereas the lagging strand has discontinuous synthesis, which has, again, the same 5' to 3' direction, but it's creating small little 5' to 3' segments called Okazaki fragments. Because again, the leading strand, all one giant strand, lagging strand, little bits and pieces that vary in size. They can be small, they can be a few hundred bases, they can be a few thousand bases at a time. It just depends on the organism. They vary in size. But typically speaking, they're generally relatively large, like 1,000, 5,000 bases in their bulk. But it creates on the lagging strand a bunch of these Okazaki fragments, which have to be put back together, and we'll get to there in 1 second. Next up, we have DNA polymerase 1, which has to come through. So it's a different class of DNA polymerases. It comes through, removes these RNA primers. So only one on the leading strand and a crap ton on the lagging strand. Every time the lagging strand jumps back, remember it has to synthesize a new primer. You looked at me like, so it has to remove a bunch of these RNA primers because DNA has to be, only DNA, can't be RNA and DNA. So it has to move the RNA primers, and that's the job of DNA polymerase 1. And then we have our final enzyme, DNA ligase, which comes together and synthesizes the lagging strand together, so it knits together all of our Okazaki fragments, thus creating our model of semi-conservative DNA replication, which is exciting. Now, when we're talking about this, if it's a linear strand of DNA, that's cool. Right? When we're talking about your chromosomes, this model works perfectly. There's no kind of weird things to think about. But when we start to think about our prokaryotes or our mitochondrial genomes or chloroplast genomes, we have to actually start incorporating these circular pieces of DNA, which is how our prokaryotes and the ancestors of our prokaryotes, the mitochondria and the chloroplast, house their genomes. Now, in the case of our circular genomes, they actually proceed bidirectionally from a single origin of replication. So you have one way, it goes completely in one way circle around, so it does one full leading strand, and the other way goes a completely lagging strand. So we have this bidirectional way of replication. You get this kind of weird distortion that occurs here, which ultimately has to be held together. Ultimately has to be fixed by an enzyme called DNA topoisomerase. So it relaxes supercoils that form. But what you need to know is that replication in prokaryotes, and maybe this wasn't super clear from the slides, it goes one way, it goes the other, they meet together, 2 brand new circular chromosomes. That's all I really care that you know. Because again, what we talked about before was just a completely linear section of DNA. But when we have circular DNA, we have to take into account the circular aspect of it. We can't break it into a strand. It has to stay together. And how does it stay together? Well, bidirectional replication. I just want you to know that's a thing as opposed to knowing every nitty-gritty stuff, which is why I didn't dive into it super deep. Okay, so inheriting your DNA is important, but accuracy of the DNA is probably arguably just as important, right? If I have a trait I want to make sure I pass on that trait and not passing on some version of the trait or potentially a non-functional version of that trait. So there's 3 ways to ensure fidelity or accuracy of your DNA. So we have redundancy, which means either strand of the helix can serve as the sequence for the other template. So if you have a mutation in one, the other one has the correct sequence. So that's the level of redundancy there. There's the precision of cellular replication machinery. So DNA polymerase 1 and 3, which we just discussed, have proofreading ability. There's a ton of this in chapter 7 in your book, just as a note. If you want to like really deeply dive into it. I usually don't like to reference the textbook, but it's actually, there's a lot of good stuff in your textbook if you're not reading it. Just as a note. And DNA repair enzymes, again, also described in chapter 7 of your book, also help fix any problems in your DNA. So there are 3 mechanisms by which you ensure the accuracy of your genetic material from one generation to the next, or one cell line to the next, right? Liver to liver. Right? So let's talk about recombination. It's an important part of genetics. Genetics, or genetics, not genomics. Now, as we've mentioned previously, new combination of alleles in gametes are created by 2 distinct events in meiosis, 1/2. Independent assortment, which we covered in a previous lecture. It's also in chapter 4 of your book. Each pair of homologous chromosomes segregates freely from the other. So chromosome 1 isn't reliant on chromosome 2 or vice versa to segregate. They just do their kind of their own thing. Every cell gets the correct numbers, theoretically, but they're still inherited independently. So no chromosome is dependent on another. Now, within a chromosome, the genes are dependent on one another, right? Right, as we discussed with linkage, right? The closer you are, the more higher probability that you're linked. But generally speaking, most allele combinations are unlinked, especially if they're on different chromosomes. Crossing over, again, as we've mentioned and covered in chapter 5 of your textbook, does create new combinations for linked genes. So as we discussed last class, recombination or just pieces of DNA just swapping places, right? Creates new combinations, new assortments of alleles, which gives rise to more genetic diversity, which is an important thing. So, as we discussed previously when we talked about briefly about tetrad analysis. Now, when we looked at tetrads, tetrads result from recombination after the chromosomes themselves are physically replicated. Now, we can look at with this. We have duplication. So we have A. It's A. I don't know if you guys can see the A's on the slides. Even like I stand here looking at it, it's blurry. But in AB, we have a recombination event that happens during meiosis 1, leading to a shuffling of these alleles. So we have A plus B, A plus B plus, and then we have a shuffle, right? You just tell by the changing colors here, right? And then meiosis 2 causes those to be segregated into distinct distinct gametes. So again, not too much crazy. Now, tetrads that have recombinant progeny contain an equal number of both classes of recombinant. So remember, when recombination occurs, we can I'm going to skip this because I forgot what I was going to say. But the model here, again, what recombination looks like, how it brings together brand new combinations of alleles. I'll think of it like at the end of class. So let's talk about this idea of recombination. So Messelson and Wiegel, so you guys might remember Messelson from one of our previous slides in this class. They actually co-infected E. coli with with an AB and an AB phage. So big A, big B, and big A, little b. So they infected E. coli with 2 distinct phages, 2 distinct genotypes. So they grew the phage on heavy carbon and heavy nitrogen, so creating the big A, big B as heavy, and the little a, little b as light. as you can see, just growing on 12C and 14N. So just a regular garden variety nitrogen and carbon. Now, the idea here, if we're combination between the genomes that has not replicated during connection, so if there's no replication of genomes at all here, we would have a mixture of heavy DNA and light DNA, and light DNA and heavy. So mixture. So you can see recombination happening in real time by a mixture of heavy and light DNA. And if DNA has to break and rejoin, we expect to see some AB phage with mostly heavy DNA. And we can, again, separate this out by our friend centrifugation here, which is cool, right? So this is kind of how they teased out how recombination was working, right? Using these heavy and light, well, I should say just normal forms of nitrogen and carbon. Remember, our DNA bases are just all nitrogen and carbon, right? There's oxygen too, but it's mostly carbon and nitrogen. So. Now, recombination at the molecular level actually is mediated by a really heavily conserved protein called SPO11, and it's kind of a remarkable conservation here. It's found almost in every lineage of organisms, which is really incredible. And I say that because the only genes that are really found on that level of conservation are like core machinery, like glycolysis and oxidative phosphorylation and DNA repair, like core, like central things that every cell needs. And this is that's kind of a big deal, right? It's found across so many. And so we can describe recombination as what we call the double-strand break repair model. And so let's talk about this model, how recombination occurs. So going slightly more in depth than we did in a previous class here. So step one tells us SPO11 breaks phosphodiester bonds. So it acts essentially like scissors, which is why it's obviously as scissors here. And we have two non-sister chromatids here. So that's the first aspect that SPO11 catalyzes. Step two, which is called resection. Now the 5' ends of each strand are broken, and they're degraded by an exonuclease, creating what we call 3' single-stranded tails. So you can see we broke our DNA here, and then we have this exonuclease. Anytime you see exonuclease, it just simply means that it's just essentially degrading pieces of DNA. That's really all it does. I know it sounds like a complex word, but it's just really just degrading DNA. Anything that's a nucleus is just degrading DNA or RNA. So we create these 3' single-stranded tails. Step three, which we call strand invasion. We have another protein which we call DMC, not to be confused with run DMC. But DMC1 helps one single-stranded tail invade a non-sister chromatid, forming what we call a heteroduplex. So what we're looking at here is we started with this non-sister chromatid here in blue, and we have this RPA, which again, we have DMC1, which helps this essentially break apart this other non-sister chromatid here, allowing for this one to essentially come in and bind, creating again two pieces of single-stranded DNA here and a piece of single-stranded DNA here, allowing red to essentially hybridize with blue. Now, just as a note, recombination only works when there's a decent level of homology. Just as a note, right? Combination doesn't work on things that are very, very divergent, because for something to bind like this, it has to be similar. Just as a reminder, it has to be relatively similar. So it can't just be like our chromosome and bacterial. Right? It has to be pretty simple. Step four, which we call the formation of a double holiday junction, which was named not after like it's Christmas holiday or anything like that, just a person named Holiday who discovered it. Pretty cool last name. Now, so we have this process here. Now you'll notice that we have the ligase is still present, right? It's keeping this part. We're forming this brand new, essentially, aspect, which we call this D loop here. We have the original, so we have blue strand is invading red strand, and red strand is invading blue strand. So it forms this, essentially this loop. So we have a heteroduplex, but this whole process here, what we call a D loop. Now, DNA synthesis then comes in. So we have, another polymerase coming in. It has to replace any DNA that was degraded. So just as a refresher from a few slides ago, one of our first things was that exonuclease came through and degraded some DNA. So we have to have another polymerase come in and fix this, as denoted in blue here, and technically blue. So we have to repair that DNA as well. And ligase has to reseal the DNA backbone. So what we end up getting is this heteroduplex region where we have, again, invasion of a strand from the bottom, invasion of the strand from the top, forming this heteroduplex, which is, again, just a combination of blue and red. And this heteroduplex region, once it is formed, it's actually elongated and extended out. So basically, you can kind of think of like you have two strands, and they're basically like intertwined like rope, and they're just you're just pulling them together to create this sort of like elongated area of recombination. So how do we finish all this up? Well, we elongate, and eventually we either stop elongating because things are just not similar anymore, or something stops it. But we have to resolve these holiday junctions, right? These intersections between the top strand and the bottom strand. And the reality is they're just resolved by DNA ligase. Ooh, that should sound familiar, right? Our friend DNA ligase. So he should come through, nip this in the bud, separating these two strands, resolving these holiday junctions, giving us two distinct strands of DNA that are a mixture of our original red and our original blue. And again, you can see if we think about this as genetically speaking, well, if we started with capital A, capital B, well, now we have capital A, little b. And if we started with little a, little b, well, now we have little a, big B. So mechanistically, this is what's going on in recombination. Now, what's important for you to remember? Well, the reality is you mostly want to know what crossing over is doing. Swapping bits and pieces based upon homology. Right? That's how replication actually physically works. And again, visually speaking, from a genetic perspective, this is how recombination leads to the organization of the bringing together of new alleles. Right? If I had dominant, well, now I have dominant recessive. Maybe that's a completely new set of traits. So conceptually thinking about that is ultimately what's important for this scenario. Now, recombination can also occur without crossing over, which I think is a pretty cool aspect. Now, anti-crossover helicase, which is kind of a loaded word, but it's a helicase, so it's preventing crossing over, right? That's all it breaks down to be. What it does is disentangles the invading strand from the non-sister chromatid, preventing the formation of a holiday junction. So we're not getting this forming. It essentially says, well, it basically just goes, nope, I'm not going to let you invade. We're not forming any holiday junctions here. But we still have these open strands here. And DNA ligase has already degraded this region, right? Because we have these open strands. Now, when we have this, let me take a second. So we have we can have this happen. I should say. So we can have this process happen, but the helicase is essentially halted. So it doesn't have this fully formed set of holiday junctions, but you can get a minor level of crossing over without any meaningful amounts of recombination if you have just a little bit of invading here as you go. Just as now, DNA, two strands of DNA that simply don't break and rejoin at the same location. I'm gonna skip this because I can't remember what I'm listening here. All right, so let's move on. Sorry, my brain is not remembering all the things I need to say about these things this morning. I apologize. Which is why I don't write the exams until after I've given the lectures. I know what I've made a mistake on, and I don't ask you questions on it. Now, recombination is great, but as you can probably reason, these heteroduplex units, well, that's kind of a dangerous thing. Right? What happens if there's mismatches? What happens if recombination occurs and it's not as similar as you would like, and you get A binding with C or T binding with G, right? That level of mismatch. And so we can have mismatch, which we'll talk about in a bit. So there's DNA repair enzymes to eliminate any mismatches, right? G with A. But we can have what's called gene conversion, which is kind of an interesting phenomena, which is basically the physical exchange of one allele for another in a heterozygote. Sorry, the physical change of one allele in a heterozygote to the other. And again, this is all based upon this mismatch. So if we have G binding with A, well, that has to be resolved, right? Well, what happens if we resolve this to a T, and now we have T, or we resolve this to a T, and this we resolve to a G? Well, gene conversion actually tells us that, well, if resolving it in the correct way might actually induce a brand new set of alleles. So you can imagine, like, if the trait is close enough genetically that one base pair might actually matter, well, you could pretend that, well, maybe you could originally coded for, say, in this case, if you coded for big B, well, maybe that single base pair changes it to a little b, or vice versa. Right? So that's what gene conversion is, right? So again, in theory, recombination should only occur in areas of high homology. However, as we're seeing here, single base pair matches can occur. And as you guys probably recognize, single DNA-based mutations are kind of a big deal for many genetic disorders, right? Sickle cell, cystic fibrosis, right? Single base pair changes, big changes in phenotype. So in this case, we can actually have the brand new conversion of a big B to a little b, or a little b to a big B, simply by using our DNA mismatch repair system. Again, identifying a G is bound with an A or a T is bound with a C, and fixing it to the way it should be, potentially changing the genetic code, which I think is kind of a weird thing. But you have this really important event of recombination, and they're bringing together new alleles. But maybe you bring together new alleles, and then you just bring out a deleterious one just because you're trying to fix a mismatch here. Seems kind of, kind of silly, right? So, anyways. I'm going to skip this slide because I don't think it's that important. Now, in contrast with the random nature of homologous recombination that we just discussed, we can also look at what's called site-specific recombination, which essentially promotes the breaking and then rejoining of DNA molecules at very particular areas of the chromosome. So this is catalyzed by a single protein, aptly named recombinase, enzyme ASE. What does it do? Site-specific recombination. And this essentially is specific enough to catalyze all breaking and joining steps. So previously there was a number of different molecules present, right? Different proteins that were doing different functions. Well, luckily for us, there's one enzyme that does all of this for us. Now, the idea is if we have this very specific location, so again, the site-specific recombination that we want to occur, what do we have to have that happen? Well, step one is cleavage. So we just have to snap it, right? Cleave, like a meat cleaver sort of thing. We have the strand exchange, so we have to have the swapping. And then we have to have ligation, so they have to bind together. And then we have a process which is called isomerization, which, I don't know, kind of doesn't look all that crazy different. But basically, just we have a binding together. And we're starting to have the strands essentially elongate, coming back together. And then we have the final part, which is cleavage, leading to, ligation, which leads to the strands essentially breaking apart. And again, all catalyzed by a single recombinase. So again, specific sites, only small levels of holiday junctions forming. So not, just as a contrast, not this double site that we have here, like over this large stretch. Again, very small stretches of DNA that we're going for here. Now, bacteriophages use site-specific recombination to actually physically integrate their genome into the host bacterium. Viruses outside of bacteriophages do this as well. So DNA-based viruses, they basically stick their genome inside your genome, and they do it by site-specific recombination. So you guys have probably heard, well, maybe you haven't. You might have heard the sort of the term about 7% of the human genome is virus. Well, why is that? Site-specific recombination. DNA-based virus have incorporated their genome into ours and just kind of hung around. Now, let's look at this. Actually, I just want to make one mention of one other thing. Now, this is a cool thing. So it's the bacteriophage sticks its genome inside the host. Now, it has to replicate its genome from that, but it also means it has to get, it has to essentially excise its genome from there. And kind of interesting, you can actually use it to reshuffle regions, use this idea of site-specific recombination of a bacteriophage to actually shuffle things around inside the phage's chromosome and the bacteria's chromosome as well. It's kind of a cool thing. So it can artificially induce some genetic novelty into itself. It's kind of a weird thing, but it does. But ultimately what we have here is integration, where we have physically integrating of the bacteriophage's chromosome into the host chromosome. The bacteriophage is designed here by purple in a thin line. Host chromosome is this dark black line here. Then it has to physically be excised, so basically breaking the phage's chromosome out of the bacterial chromosome. And then we have to have an inversion of the chromosome. So basically, you see that the phage's chromosome is partially red, partially blue here. It has to be re-inverted back in. And then once it's reverted back into the host, it can actually have a recombination event inside the host chromosome. So again, I don't anticipate you guys to remember this stuff. I just think it's a cool aspect of recombination. Provide some novel tools for phages, novel tools for bacteria to use for extra levels of genetic diversity. Now, biotechnology-wise, we actually use recombination quite a bit. So there's many different techniques out there, including fragment length polymorphism, to manipulate genomes experimentally, so we can study the effects of recombination experimentally in a lab setting. You guys also might have heard of CRISPR-Cas9. It's a bacteria's immune system, but we've adapted CRISPR-Cas9 for genome editing, and it does it by recombination. And we can use CRISPR-Cas9 and the RNA that it produces to cause site-specific recombination at a specific point in the genome of interest. So that's one of the cool things about CRISPR-Cas9. If you've never read up on CRISPR-Cas9, definitely do. It's really fascinating. But this idea of site-specific recombination is what CRISPR-Cas uses to make precise edits in the genome, like really, really accurate edits in the genome. So I'm sure you guys probably was it last year, 2019, where they had the babies that were born in China that were edited by this technology. Again, they edited out a specific region of the genome with CRISPR, which again relies on site-specific recombination. Pretty cool technology. If you don't know what CRISPR-Cas9 is, you should. It's one of the futures of biotechnology. If you ever have any desires to work across the river, you should know what CRISPR-Cas9 is, how it works. All right, so part 2 of the lecture is going to be focusing on mutations. Now, you guys know mutations are inheritable changes in the base pair sequence of your DNA. So it has to happen, in our case, it has to happen, mutations can occur in your somatic cells, but we're thinking about inheritable ones. Well, it has to happen in your germline cells, right? Bacteria just has to happen in their chromosome. Now, we can have what's called a forward mutation, which changes a wild type allele to a different type of allele. So going from red eyes and Drosophila to, say, a mutant white eye type. So, the example I have here is A plus to A. Remember, plus always tells us the wild type, or B plus to B, right? Or we can have a reverse mutation, or what we call a reversion, which changes the mutant allele back to a wild type. So little a to A plus or B to little b plus. So two distinct types of mutations in a genetic context. Now, when we're thinking about genetics, we really are thinking about things that are heritable, right? We don't really so much care about things that happen in your foot cell, right? We want to know how things are inherited, right? That's what genetics really is. So, excuse me. Now, there are many different classes of mutations, and we're just going to go through them relatively quickly, because these are things that you guys have heard multiple times. But we have substitutions where we replace one base by another, which can lead to one of two different things: a transition, where a purine is replaced by another purine, or a pyrimidine is replaced by a pyrimidine, or a transversion, where a purine is replaced by a pyrimidine, or vice versa. So that's our substitutions. We can have deletions, where we block one or more base pairs from insertion. So you can imagine, if we remove a piece of DNA, in this case, you have 3 bases, it's completely removed. That's a deletion, which is in contrast to an insertion, where we stick in DNA bases, and they're variable in size. So you can do a 1 base pair deletion or insertion, or they can be megabases. And what does megabases mean? Mega meaning million. So deletions can be monstrous. Insertions can also be monstrous, millions of bases long. So we like to think of it in the most simple context of like 1 or 2, maybe 3, but again, they can be quite large. A lot of evolution happens because of large deletions. Definitely. Now, point mutations, as you guys probably remember, where we affect one or few base pairs, altering one gene at a time. So when we have transitions, transversions, we also have small deletions or insertions, are all what we call point mutations. So basically what we just talked about on the previous slides. Now, mutations can also affect multiple genes via the reorganization of a genome. So changing the synteny or the order of the genes is kind of a big deal. We'll cover this in when we talk about chapter 14 in maybe 4 or 5 weeks. But just kind of a foreshadow. And we can actually look at some example of this. So there's an ongoing study right now in mice where they look at rates of forward mutations at 5 coat color genes. So there's A, B, C, D, and LN. So not completely unknown to us in terms of letters to denote genes. But a given gene mutates to a recessive allele basically in 11 out of every 10 to the 6th gametes produced. And it varies. So the number of mutations, the number of gametes tested, giving you these mutations, right? At about an average of 11 per 10 to the 6th gametes, giving us a mutation rate in mice, of course, about 10 to the minus 6. So from an evolutionary perspective, from a genetic perspective, not too common, right? If you have to produce 1 million gametes to get 1 mutation in a gamete, that's not very common. And the rates of mutation in somatic cells are equally as uncommon, 1 in a million. Recombination, much better, right? Crossing over, way better. Independent assortment, way better, but not too common. And so while we classically think of mutations as being the way we get new traits, again, not super common. However, if you think about this in an evolutionary time frame, well, maybe that's a little bit more digestible. Right? When we think about life has been on this planet for billions of years, well, a 1 in a million chance is pretty good over a billion years, or even 100 million years. Not on our lifetime, but in an evolutionary. Just as a note. And different genes have different mutation rates. Some genes have very, very, very low mutation rates, so 1 in a billion. Others have relatively high ones, 1 at 1,000. So much more common. And it depends on the gene size. Now, if you guys were to make an educated guess, what type of gene do you think mutates the most? Small genes or large genes? I heard large and small. So I heard large. Yeah. If you have a 1 in a million chance, say if you have a 1 in a million chance of throwing a dart and hitting one of these letters here, well, the letter's bigger, so we can have a greater chance of hitting the letter. Right? If it's bigger. Same thing with your genes. Bigger genes typically mutate faster. And some genes are just more probable. And it's just depending on their function. So genes that are really, really important, they think about metabolic genes, they're much less likely to mutate. Whereas opposed to pieces of your genome which really don't do too much, they're likely to. So it just depends. The average mutation rate in gamete-producing eukaryotes is higher than that of most prokaryotes. That's just as like a background mutation rate. There are far more mutations that occur in microscopic organisms than macroscopic organisms, but at a background rate, they're higher in eukaryotes. And why is it higher in prokaryotes normally? It's because they try to mutate themselves. Just that's it. Remember how I mentioned DNA polymerase 4 earlier in the lecture? That's a DNA polymerase 4 thing. Targeted mutation. It's a cool thing. Now, human sperm also have a higher mutation rate than human eggs. Now, mutation rate is about 1 in 10 to the minus 8. So each child has about 60 mutations. So base pairs that are different from either parent, which I think is kind of a wild thing to think about. Right? I don't know about you guys, but I look really different from my parents, both of us, not just my mom or my dad. But I look pretty different, and think that there's only 60 base pairs of mutations that have occurred that partially give rise to that. I mean, recombination is the other part, but 60 base pairs of mutations, not a lot of mutations every generation, right? Kind of a weird thing. And most mutations, as you'd imagine, simply don't affect phenotype. Most of your genome, remember, the coding capacity of the human genome is only about 3%, i.e. 3% of genes, 3% of your genome makes a protein. Most of your genome doesn't seem to encode for anything. So trait-wise, a very small proportion can actually make a trait. The rest of it kind of doesn't do too much, just as far as we know. Now, the mutation rate in sperm varies between 2 to the minus 4 and 10 to the minus 8. I'm sorry. Yeah. I'll leave it at that. Now, why is that? Well, simply because males produce our sperm continuously, whereas females, they only had one shot at producing their eggs. Right? All the eggs that you ever had were produced before you were born. You lose about half of them when you go through puberty, but all the ones that have ever happened were produced a long time ago. Well, relatively speaking. Whereas males, we do it all the time. Constantly. So, and mutations also are more likely to occur in sperm from older fathers. So if you ever see like an 80-year-old guy having a kid with a younger woman, he's got old mutated sperm. Gross things about that. Just thought I'd make mention of it, since we're like an hour deep into this. You guys look like you're ready for it now. So now, as we know, mutations can disrupt gene function, and very few have been shown to ever restore gene function. So there's an example here. We have a rare reverse mutation. So we have 2 albino mice, so the lack of pigment, and we have a rare reversion event where we have a black, or I should say probably a brown mouse. And why is that? Well, there was a reversion event here. GC was reverted to TA, causing the switch in the genotype and the phenotype.
So in that case, that mice is its own. It's that mice, but it's not considered. Is that normal? Like when it does that swap, is it bringing it back?
It's bringing it back to it, because brown would be a wild type, as you can maybe see, it's plus. Mm-hmm. Whereas the albinism gene, AL minus, is not, is a mutant type, right? So technically reverting it back to a wild type. Right. You can imagine, though, it could go the other way, right? We go from wild type, maybe this was wild type, now we're going to mutant. So we have a reversion here. But typically speaking, when we think of a lot of mutations that affect progeny, they're usually deleterious. Not all of them, obviously, but typically speaking, we don't see a lot of restore of function mutations. So this leads us to looking at some evidence that mutations occur in bacteria, not directed, but do occur spontaneously. this is shoot, I forget his first name. Luria and Delbert. Now, Luria is a pretty cool scientist. Those of you that have ever grown any microorganisms with, say, Dr. H or myself, you grew on Luria bertani media or LB media, named after her. She developed it, just as a note. And they examined the origin of bacterial resistance to phage infection. Because, remember, viruses attack surface proteins. If a mutation that changes the structure of the protein, the virus might not be able to recognize the protein. Right? So thus you have resistance. Now, what they were doing was infected wild-type bacteria with phages. Interestingly, though, the majority of the cells were dying, but a few actually were able to grow and divide. And the question you could ask are, had the cells altered biochemically? So is there something adaptive about the phage being present? You could ask, did the cells carry heritable mutations for resistance? Or did the mutations arise by chance, or did they arise in response to the phage, right? So phages being a selection pressure, right? So thinking about Darwinian evolution. So let's talk about this. So we have two hypotheses here. Resistance is physiological, so it's adaptation, essentially. And what you'd imagine that essentially the pedigree of bacteria would look very different depending on how this worked, right? So if resistance is physiological response, you would expect to only see it in younger generations because it's something that they would have to adapt to over time, right? Biochemical thing, maybe it just requires them to build up a chemical over time. You would expect to only see it in later progeny. Hypothesis 2 tells us, well, resistance arises from point mutation. And we would expect to see that in a couple different ways. But ultimately, we would likely see it occur in an ancestor. So back here in the pedigree, there we go, and then being inherited by the ancestors. Right? Which clearly this one is the correct answer. But again, two distinct types of pedigrees to parse out between the two. Now, conversely, you could also think, well, maybe it's fluctuation, how long they were exposed to that viral agent, right? The evolutionary thing. Well, thinking about fluctuations of the number of times they were exposed to the virus. And you can imagine, like, the more times you're exposed to the virus, well, theoretically, the more likely you are as a population, because remember, individuals can evolve, but as a population, you're more likely to evolve resistance to this. So if you have multiple exposures, you would expect through time, after multiple exposures, to see more and more colonies. But the reality is you just don't see that, right? It's random. There's 170 colonies, 107 colonies here, implying a high level of resistance. There's 0 colonies, implying no resistance. So what we learned from this experiment, which I think is pretty cool, is that, well, it's not biochemically, and it's not evolution, but it's actually just a random mutation event that's occurring. Now, this should make sense from thinking about Bio 2, right? Bio 214 that you guys took. Well, a mutation popped up that was beneficial from an evolutionary context. Now, what is that context? Producing offspring. If it makes you better at producing offspring, like this individual would, you'll produce more offspring, and your offspring will have your traits that made you more successful at producing more offspring. So this is like a strict evolutionary pressure. This is, again, thinking about evolution too. Genetics and evolution are tied together 110%. Just that's it. And you can also do what's to looking at resistance as well, but we're actually going to skip this over because it's the same thing, just told in a different story. So how do we interpret Lurie and Delbrick's results? Well, bacterial resistance arose from mutations that occurred before the exposure to this bactericidal event, in this case, a virus, or you can think of it as an antibiotic as well. Now, this bactericide becomes a selective agent, so in both an evolutionary and just a population numbers thing, killing non-resistant cells and allowing the survival of cells with pre-existing resistance. And essentially is what their results are telling us, that mutations occurred as a result of random processes, right? Now, once random mutations occur, they usually remain stable, unless they're really, really deleterious. Right? You can imagine if it's really deleterious, you can't use glycolysis anymore, you're going to die. But if it's something minor, like being resistant to a phage, well, it's kind of come on over time. We see this a lot in bacterial-based resistance to like antibiotics and stuff like that. So for instance, like tuberculosis, we used to use rifampin to treat tuberculosis infections up until the 1980s. And then a population of tuberculosis arose very much the way this one did, that just happened to have a random mutation that made it immune to rifampin, which led tuberculosis to come back and be resistant to rifampin. We use other antibiotics now, but this idea, again, might seem abstract, but again, this is how we know antibiotic resistance works a lot. Random mutations that might not seem beneficial at the time, but they're kept because there's no real cost to fitness or the biology of the organism itself. Now, there is this really interesting race between DNA repair and DNA replication. Now, base pair changes are often corrected by DNA repair mismatch systems. So if I have an A that changes to a C, well, my mismatch repair systems, as we saw previously, come and say, nope, that's not right. I'm going to fix it. So it corrects that mutation. However, after replication, both DNA strands contain the base change and are inheritable results. So if I have a change that's not picked up by a DNA mismatch repair system, well, then I can have essentially that being passed on to the next generation. So it is a balance between the two. Your mismatch repair systems are great, but they don't catch everything, which is how we can have 80 mutations relative to our parents. So it is a balance between DNA repair and mutations that occur. Just as a Now, there are natural processes that induce mutations through DNA damage. So one of which we're going to talk about is called depurination, which is the hydrolysis of a purine base. Now, you'll notice that it's 1,000 per hour in every single cell, which is huge, right? I told you 10 to the minus 6. This is 1,000 per hour. You have 38 trillion cells, which means this happens what? 38 quadrillion times in your body every second, every hour. Crazy common, right? Now, what is depurination? Well, hydrolysis. What's hydrolysis? Does anybody remember from chemistry? You can kind of piece together what's going on from here. Well, it's involved with water, right? So we're essentially cleaving guanine and replacing it with water. HOH is water. That's all we're doing, right? Hydrolysis is essentially adding water, right? So that's what's happening. So cleaving, say in this case, it's just guanine, but we're creating what we call an apurinic site. Again, where we have no G, right? There's no nitrogenous base there. Or we can have what's called deamination, which is less common, but very few things are as common as depurination. You good? Which is the removal of an amino group, which is NH2. So in this case, we have cytosine. It has an amino group. It's deaminated, so we remove this, creating uracil, which is wacky and wild. We're going through DNA and RNA here. Interestingly, though, you'll notice that the 5-carbon, the DNA base isn't all that different, and the sugar here would be the same as well. So it's kind of this weird mutant, right? But again, we have normal sequence CG. Deamination occurs, creating UG. Replication occurs where that is propagated through time, creating eventually a mutant sequence, which is weird, right? You have U, which inherently tells you it's wrong. So your DNA mismatch repair should come in eventually and say, that's a U, that makes no sense. I'm going to fix that to, well, it's supposed to be an A is on the other side, so that should be a T. But we know that, well, it was originally C and G, right? Kind of this funky little system. But again, normal processes in your body creating single point mutations. And I say your cells, I say your body, I think your cells. Now, cosmic rays, so sunshine and X-rays, naturally speaking, and from doctors and airplanes and all that fun stuff, break the sugar phosphate backbone of DNA. So I'm sure you guys have heard that before. You know, ultraviolet light and dangerous stuff breaks your DNA. Breaking your DNA might cause a deletion if it's not properly repaired. So that's what we're seeing for X-rays here. So when you see like the X-ray tech run behind the safe door, he's trying to prevent this from happening. And you know, getting blasted with X-rays once is cool, not really that big a deal. But getting blasted with X-rays 10 times a day for years and years and years, clearly not so good.
So I work at an animal hospital. So when the robes and all that gear that we have to put on, what would be in there that like makes it so heavy, that makes it so?
It's usually lead.
What is
Usually
it?
lead.
It's lead, right?
Usually lead. So I don't know if you guys saw Chernobyl, the HBO like miniseries. What they were trying to, you know, oftentimes they store radioactive material. They covered it in lead. Because lead does a really good job at kind of shielding you from not all, but a decent proportion of high intensity radiation.
Because when we do the X-rays, we have to do like a whole head to toe dress with the heavy robes.
Yep. So yeah, that's it's trying to prevent. And they also have the special screens to go behind, again, preventing the X-rays from blasting you and doing this. Now, interestingly, ultraviolet light produces what we call thymine dimers. And so we have a normal sugar phosphate backbone here. A thymine dimer, so we actually have a linkage here between two adjacent thymines together. So in this case, you can see the thymine dimer is just two covalent bonds that form between two adjacent thymines together. So you might ask yourself, is TT really common in your genome? The answer is yeah. 3 billion bases, TT is going to be there quite a bit. So when you're exposed to UV light, thymine dimers are relatively commonplace. Now they can be repaired, so there's a whole mismatch repair system dedicated to thymine dimers. But what ends up happening is if you have a sequence where you say have CG, you have some sort of oxidative damage, and that oxidative damage is carried through time, potentially mutating something from GC to TA. Because again, these thymine dimers can form either TT, or they can actually just cause the mutation to occur, changing it from say a C to a T.
And these mutations can affect your health over time. And when you have kids, right?
If it's in your germline, right? So if you know if it's in your egg cells or if it's in my germline, using sperm or something, yeah, potentially has the capacity to be inheritable. Conversely, if you think about just cancer,
right,
the mechanisms we're talking about in these past couple of slides, this is how cancer arises. Right? These are how mutations occurs at the genetic level. So cancer is like kind of the phenotype you see is like bam. But you can also think there are weird conditions where like genetic disorders where you typically have to inherit from your parents can arise because of these. So Huntington's disorder, for instance, can arise via these processes as well, where you usually have to get it from your parents, but there is spontaneous acquisition of some genetic disorders because of this. Not super common, but it can be. I say that because Huntington's disease scares the crap out of me. It's scary. So, now, we can also have oxidative damage. Now, what does oxidative damage really refer to? Well, it's oxidation, right? So the loss of electrons, right? That's what we're seeing here. Now, what happens is you have an oxidation of our ring. So we're losing electrons here. Oxygen is gaining them. The carbon ring is losing them. So there's an oxidative event. Now, what causes this? Well, active or reactive oxygen species. Now, you might say, what's a reactive oxygen species? Well, hydrogen peroxide is an example of a reactive or active oxygen species. It's anything that has the capacity to be catalytic. That's all it really means. So why would that be a really common thing? Well, the reality is your metabolism produces lots and lots and lots of reactive or active oxygen species, which is kind of why your mitochondria exists, right? You don't just do metabolism in your at least the most destructive forms of your metabolism inside your cytoplasm. You do it in your mitochondria. And why is that? Because you produce lots of these active oxygen species, which in turn can damage your DNA. Taking guanine, again, changing this hydrogen to a double-bound oxygen, which again, you can see we're changing this pretty dramatically chemically, which creates GO, or 8-oxo-DG. So it's guanine, but not quite. It's oxygenated guanine. Again, distinct type of guanine, not going to quite function as guanine. So again, these are all just spontaneous mutations that happen to your cell. Really, really common. Oh, I guess I say really, really common, relatively common, right? Now, you don't have to have random mutations. You can have errors during DNA replication. So as DNA polymerase is doing its thing, well, it might say, oh, I have a G here. I'm going to add accidentally an A. DNA polymerase 3 is really accurate. It has an error rate of 10 to the minus 9. So 1 in a billion, which is rare, right? 1 in a billion chance. If you were to tell me that I had a 1 in a billion chance of, I don't know, being hit by a bus, I wasn't really worried about being hit by a bus, right? But if you told some other people you got 1 in a billion chance of winning the lottery, they play that lottery every day. Right? I had this conversation with my mom last night. I was just making fun of her vicariously through you guys. So, anyways, so it's pretty rare. Now there are error-prone polymerases. DNA polymerase 4, which I've mentioned multiple times this lecture, is an error-prone polymerase. It has an error rate of 1 in 100 or 1 in 1,000, so it's designed to make mutations. But 10 to the minus 9 is rare. But the reality is DNA polymerase has proofreading capacity, meaning it can go through and figure out what's going on. It could say, okay, well, I put the wrong base in here. I can then go excise it and pull it out and replace it with the right one. So DNA polymerase has that ability in real time. And I think it's kind of lost of how fast DNA polymerase moves. Remember, this system moves at 1,000 bases per second. So it's able to recognize over a strain of 1,000 bases, synthesize them brand new, recognize any errors as it's doing it, pull them out all like that, which is incredible. I mean, very small distances, obviously, like angstroms, right? 3.4 angstroms between bases. But again, it's really, really fast. It does it really accurately. So now, each base, we can have this event called tautomerization. I think I said that right. Each base has what we call 2 tautomers. So there's chemical form that interconvert regularly, causing incorrect DNA base pair matching. Now, let's look at what this looks like. Now, we have this rare tautomer base of we have adenine here, guanine here, right? Guanine should bind to C, adenine should bind to T. But you see that adenine here has this rare form that looks like this. Thymine has this rare form that looks like this. Guanine has this rare form that looks like this. Adenine has this rare form that looks like this. And thymine has this rare form that looks like this, which causes them to have the capacity to hydrogen bond. So when I say that there's this mismatch here, well, what's really causing it? Part of it is this. Part of it's random, right? You could theoretically have a T bind with a C because they essentially could link it with 2 hydrogen bonds. But the other part of it is that sometimes there's these rare forms, cytosine, thymine, guanine, and adenine, that just allow them to form the correct number of hydrogen bonds, which I think is a kind of a cool thing. Is this the first time you guys heard of this part? It's a weird thing, right? Just alternate forms of your standard A, T, G, and C. But remember, these are all synthesized via enzymes. Right? So there's capacity for these enzymes to make a mistake. Now, you can also so the way this works, we have tautomerization occurring. If it's supposed to be AT, well, if we have the weird T, it's going to want to bind to G, right? So if we have this weird T get stuck in there, well, it's going to go through time, and that's going to change to TA sometimes. It's going to revert back to the regular T, or it's going to be converted to it's going to recognize it as a C, and over time we're going to have CG form. Kind of a weird thing, but alternate forms of DNA bases. Throwing a wrench in Watson and Crick's little model here, right? Now we can also have trinucleotide repeats. So as you'd imagine, just trinucleotide, it's CCC or AAA, right? Trinucleotide repeats. Now these can expand or contract during replication. Now there's about 20 human diseases that have unstable trinucleotide repeats. And in this case, we have CGG, could be AAA, just the same repeating monomer over and over and over again. Now, a couple of these include Fragile X syndrome, and we've mentioned Huntington's disease over time. And it's kind of interesting, children of carriers also might have expanded number of repeats. And there seems to be, for at least these two diseases, the more of these random repeats you have in the gene of interest, the worse your disease is. So in this case, we have the CGG, there's 56 of them in wild type. In the pre-mutation alleles, there's 56 to 200 repeats. And then finally, in the disease-causing alleles, there's 200 or greater. It's kind of a weird thing, right? You have this intermediate, like, I'm heading towards having something really bad. Not quite there yet. I'm heading towards having something really, really bad. And as a note, as I mentioned, the more repeats you have, the worse it seems to be. So if I had 500 repeats, it's likely to have a worse case of Fragile X or Huntington's than, say, someone that had 250 repeats. Kind of a cool thing. Because again, these are part of encoding for proteins. And if you imagine, if you have a bunch more repeats, well, I'm adding not just 56 repeats, I'm adding 56 amino acids. So you can only imagine what that's going to do to these proteins. And we could just look at a pedigree where we have unaffected and white, heterozygote, or what we call hemizygotes in the pre-mutation. So they'd be in this category here of 56 to 200. And then we have the affected in gold or yellow, whatever color you want to call that. And you can notice that the number of repeats that there is, unaffected parent, that's 22, parent, that's the heterozygote, 82. And then you can see that, that these two
Siblings?
Yes. Siblings, they have their own families, but this group has an affected child. Well, this one had that hemizygous. And again, these regions seem to expand or contract depending on, as far as I could tell, if you could find a better answer to what I'm about to tell you, awesome. But as far as I could tell from when I was doing my research, it seems to just be kind of random, like random errors in DNA replication. A lot of times when polymerases encounter things that are repetitive, it's very hard for them. So they get kind of tripped up. But if you can find a better answer to that, then let me know. But you can also see, you can see how this heterozygote is passed down. And then you have these individuals down here at the bottom, which again, unaffected parent, heterozygous parent, but full blow-on affected with Fragile X. So again, seems to expand over time, which again seems to increase with the passing genetic time. There we go. And we can look at what this conceptually looks like. Now, trinucleotide repeat expansion, or contraction in this case, DNA polymerase essentially pauses, and the newly synthesized strand slips. So essentially, it's like gets caught up on this repeat region. Then it's hard to read. So if you're thinking about if you were reading a book and it was just the same words over and over again, it would be really hard to sort of after a while just reading them over and over again. That's kind of what's happening here. And so as you have this slip, it essentially builds up over time. So we're going from, in this case, we have 1, 2, 3, 4. Then we're going to, this essentially slipping up, increasing the amount upwards of greater than 6 here. So again, it seems to just be getting tripped up. Now the opposite, the DNA polymerase pauses, and the template strand seems to slip, and these essentially get contracted out of the genome. So complete opposite process. One gets essentially incorporated in, one gets essentially excised out. Yeah.
No, you answered it. Thank you.
Yeah, kind of a weird thing. But again, it seems like the DNA polymerase is just getting tripped up. I'm sure there's probably cool research out there that I didn't find about this, but why this exactly occurs molecularly, this seems to be the case. The DNA polymerase is slipping. Now, as you guys know, and we've sort of mentioned thus far, mutagens cause mutations. Now, H.J. Mueller used X-rays, as we discussed, as a mutagen, to increase mutation rate above the naturally occurring background level in Drosophila melanogaster. So he exposed male Drosophilas to X-rays, and what he would do is he would use this mating scheme where he would have genetically marked balancers, X chromosomes, that preserves recombination with other X chromosomes. And it was actually essentially, you would basically have this event where he would try to mutate these, and you essentially have the system where he would be able to detect these random mutagens. So he needed to keep the X chromosomes intact because Drosophila can't do its thing without its X chromosome, but he still wanted to mutate these X genes. So he was using X-ray, but these are classically called mutagens that raise the frequency of mutations above the background rate. So smoking, like, X-rays, gamma rays, chemicals, any number of things can cause mutations. But we can look at what he was doing. So he had his males XY, and he had XX females. He would dose the males with X-rays, and he would have, and the trait he would have here, these bar-eyed females. Did I put a picture? No, I did not. It's just this weird phenotype of Drosophila. You can take a peek at it. But he would have these F1 bar-eyed females, but the X chromosome would be dosed. Then what he would do, he would have individual mating. So he'd have an F1 bar-eyed female mate with a wild-type male. Female mate with a wild-type male. Now, if the F1 female had an X-ray-induced recessive trait that was lethal on the X, you would create males that would die, but bar-eyed males. If the female had no X-ray treatment, they would have non-bar-eyed males and bar-eyed males as well. So they would see the effect directly on the mutation rate, because what it was doing was essentially taking his X chromosome from dad, passed down to the females, because remember the males would get the Y chromosome from dad, which he wasn't really interested in this case. So he would take these X-ray dosed males, and they would have the X chromosome here in red. And if it was dosed, it would cause the individual to die. If it wasn't dosed, it would cause the individual to live, thus having bar-eyed males. I think it's pretty cool. I'm saying non-bar-eyed males. So using mutagens to induce changes above the background rate, in this case, creating a lethal phenotype in dosed individuals, in a non-lethal trait in non-dosed individuals. That was easier to say than I did, but that's what I was hoping. Now, when we think about how mutations work, there's a number of different ways that we can think about, but we can have what we call replace a base, which I think is a cool way of saying it. But replace a base, we basically have base analogs, or chemicals that are essentially almost identical to the normal base. So in this case, we have adenine, and adenine's replace a base is actually 5-chromouracil tautomer that essentially behaves like adenine. And there's also a 5-bromosyl tautomer that behaves also like cytosine. I'm sorry, I should have said thymine. So one tautomer that pairs with either A or G. So similar to what we saw with this tautomerization previously. Conversely, we can also see hydroxylation, which is where we convert, when they add a hydroxyl group, so an OH group. So we have cytosine here. We tack on a hydroxyl group here, creating N4 hydroxyl cytosine, which will then bind to adenine, which it shouldn't do normally. Right? Cytosine should bind with adenine, but we create this weird hydroxyl added form of cytosine, and now combined with adenine. We can also have alkylating agents. They add either ethyl or methyl groups. So we have guanine. There's a chemical, ethyl methane sulfonate, which tacks on an ethyl group, which is 2 carbons here. It tacks it onto the hydroxyl group here, getting rid of that double bond. Then making guanine now being able to bind with thymine, because guanine is now O6 ethyl guanine, or G0. You can also see deamination. So certain chemicals will deaminate, very much like we saw that which happens naturally inside your cells. Again, guanine being I have the same figure twice. Sorry about that. I have the same figure twice. It's supposed to be a brand new figure. But anyways, deaminating here, removing this, allowing it to bind with a different molecule. Sorry about that. I was like, why does this have the same slide as last time? In addition, we can have mutagens insert into our genomes, which is a process. Inserting between bases is called intercalators. I believe I've said that right. So in this case, we have proflavin, which intercates into the double helix during DNA replication. Now, what this does, proflavin causes single base insertions and base deletion. So if this case you have you hit with a mutagen such as proflavin here, it can essentially come in, weasel its way in between your DNA bases, causing single base pair insertions to occur. Now, mutagens, they seem kind of bad, right? We think about mutations in humans, we think cancer, we think bad things, right? We've talked about bad things in humans, Fragile X, sickle cell, so on and so forth. But not all mutations are bad, right? Mutations we know are the mechanism by which natural selection works upon. Now, mutations that occur in non-germ cells are not inheritable, right? So if I get cancer, well, I can't just pass my cancer down to my child. This doesn't work that way. So it has to occur in germ cells to be heritable. Now, mutations inside your somatic cells can alter your survival, can alter your fitness, right? And fitness just being how many offspring you produce, or the capacity to produce offspring. Remember, that's all biology is all about. Whether it's a virus, bacteria, us, it's all about making babies. It is. And I know that's like kind of a sad way to think about life.
No, it is. That's every species.
That's life. Well, just look at your face. You're like,
I'm tired.
Is that like all life? Oh, you're tired. I thought you were like, oh, is that all life really is? Sorry. Now, the USDA actually uses what we call the Ames test to screen chemicals that cause mutations and might cause cancers, i.e. carcinogens. So they test for, they have controls where there's no mutagen, and they have a suspension of mutant bacteria in liver cells and a potential of carcinogenic organisms. And then they plate it onto a petri dish together. Now, the bacteria is histidine negative because it doesn't have the capacity to produce histidine. Now, they see if this is a mutagen, if the bacteria reverts back into being able to produce histidine. So if the mutagen has caused a reversion. Kind of a weird thing, but that's kind of how they do it. I don't know why, but that's just where they do it. But as I mentioned, your DNA repair systems are pretty good. There's lots of ways to fix what has gone bad. Now, the reversal of DNA base pair alterations is catalyzed by a couple enzymes, photolysases and alkyltransferases. They're typically homology dependent, so you typically repair bad sections of your genome with other sections of your genome that are similar to it, like pieces that are similar. That's a faster way of doing it. And there's many other mechanisms to do it that help you essentially figure it out. And I think I'm actually going to end there. I think that's enough for today. There's only like, I think, a couple more.
There's like 8 more.
There's like 8 more slides, but you know, all this is thinking about what we sort of already talked about, just fixing breaks, repairing bases. So I'm going to stop the class here because we're out of time, but also because you guys look tired.